Okay, last topic. We're going to do ABO blood typing. Oh, it'd be helpful if I could draw things. There we go. We're going to do ABO blood typing. That squareness is what happens when my processor is slowing down. Okay, we're going to do ABO blood typing, and I'm going to try not to draw too many extraneous things that I then end up erasing, because I know you guys will get pissed off at me if I do that. So we're going to talk about what's on a blood cell and what's found in the plasma for each of the blood types. We're going to address the proteins known as the A-type surface antigen, the B-type surface antigen, and the Rh factor, which is the D-type surface antigen. D is in dog. Now, I like to draw this in a table, and it's pretty similar to the table you'll find in your textbook and in your PowerPoint. That having been said, I find that you really need to draw it out and follow along, even if it's the same thing. And this table is going to be a 2x4. I've done this so many times, you have no idea. 2x4 table. The first row is going to be the cells. The second row is going to be the plasma, plasma. The cell surface is going to have, cells have an antigen. The plasma has antibodies. And the overall pattern is that the antibodies you have are going to be opposite to the antigens you have. It is opposite. That is your rule of thumb. Antigens are opposite to antibodies in any given blood type. So here we're going to put A. Here we're going to put B. Here we are going to put AB. And here we're going to put O. And we have not addressed Rh factor yet. We're going to put that on a slightly separate table, and I will explain why later. An antigen is a protein found on the surface of a cell. An antibody is a protein generally found loose in plasma. So they both start with anti, and people conflate them a lot. They have almost opposite meanings. In order to convey the different types of antigens, we like to assign these like fake little shapes to the different antigens. So when we say you have type A blood, what we mean is you have a surface antigen that we have identified as the A-type antigen. So on the surface of the cell, there will be, let's call it circular surface proteins. So a certain percentage of the population has surface proteins on their red blood cells that we have labeled the A-type protein. In a different person's bloodstream, you're going to have an antibody that recognizes this shape, that recognizes this A-type protein. So that's why it's important to us. We don't want antibodies and antigens mixing. So bear with me, I know that's getting a little bit ahead. We'll come back to it. The B-type protein, again, a certain part of the population has a surface protein that is recognizable. It's like a name tag, right? Hi, I'm A. Hi, I'm B. We're going to make this a triangular surface protein. So a certain part of the population has this other surface protein that we're pretending is a triangle, and it's recognizable. So it's like a name tag that says, hi, I'm B-type. If you have type AB blood, you have both surface proteins. You have the A-type circular surface protein and the B-type triangular surface protein. Not their real shapes, just bear with me. Hi, I'm AB. The O-type is not another surface protein. It is an absence of surface proteins on the red blood cell. In case that's not clear, in case I haven't made this clear enough, these are red blood cells, not any other kind of blood cell. So the O-type, the O is basically a null, a zero, right? It just means absence of surface proteins. It has no name tag. It's gone. Now, As I've mentioned here, the plasma proteins, the antibodies you have in the same plasma, are going to be the opposite to the antigens you possess. So if you have type A antigens, you will not have anti-A antibodies. The antibodies are going to attack. Antibodies are going to attack anything they match. So we want a person with A-type blood to have antibodies against any other type of surface protein, except for A. So A has the surface antigen A, but it has the anti-B antibodies. An antibody is going to be a Y shape in general. It's a Y-shaped protein, and they all have something called a variable region. And this variable region, for the purposes of this extended metaphor of shapes, this variable region can have different shapes. So an anti-B antibody could have triangular receiving ends. I don't want to say receptors, receiving ends. An anti-A antibody might have circular receiving ends. And we'll get to anti-D in a second. So an A-type blooded person has the A antigen, but the anti-B antibody. And they'll have a number of these floating around in their plasma. We'll keep going with this trend. If you have the B-type surface antigen, you don't want the anti-B antibody, you want the anti-A antibody. So you're going to have antibodies that look like this. The variable region will receive the A-type antigen. They will fit like puzzle pieces. Now think about it for a second. If you have both A and B antigens, can you have anti-A or anti-B antibodies? The answer is no, you cannot have either. So this not applicable. They do not have any antibodies against surface antigens, either A or B. Now, if you have O-type blood, can you have anti-B antibodies safely? Yes, you can. Can you have anti-A antibodies safely? Yes, you can. So somebody with O-type blood will have both anti-A and anti-B antibodies, which puts us in a precarious situation for blood donations. When donating blood, I want you to consider the red blood cell of the donor and the plasma of the recipient. Now, I don't think you have blood type cross-matching in your learning objectives. You do have blood type donating in your, or typing in your learning objectives. So that having been said, you might not need to do the donor-recipient thing, but there is going to be an overlapping concept when we start doing blood type testing at the end of this, how that blood type test actually works. So let's do this really briefly, and just to a level that you need for your blood type testing. Now, what I mean by these arrows again is that when you consider donations, you only consider the cells of the donor, and you only need to consider the plasma of the recipient. So what I mean by that is a person with O-type blood, if you put this cell near these antibodies, you are fine, because there is nothing for these to attach to. So this is fine. So type O donor can give to type O recipient using these labels. Type O can give here because there's no antibodies to interact with it. Type O can give here because it doesn't have any A antigens. And type O can give here because it doesn't have any B-type antigens. So O is known as the universal donor. O can give to anybody. But what happens when you put something that is incompatible in together? What if these surface antigens meet anti-A antibodies. What happens is a very specific reaction. So here's a red blood cell with a, let's say, A-type antigen. It doesn't matter if it's A or AB for this purpose. And here's another red blood cell with surface A antigens. And here's the anti-A antibody that found them. It binds to this surface antigen and this surface antigen, and it holds them together. And this is happening at a very, very large scale, and it's happening very, very quickly. And what happens is, it's called agglutination. Which means the red blood cells are clumping. I've only drawn 2, but assume this is happening on the scale of millions. So that's a really destructive thing to happen. We do not want a cell with a type A antigen to go into the plasma of somebody with anti-A antibodies. So a type A person cannot donate to a type B person, and an AB person cannot donate to a B-type person. In fact, if you look at it, can AB go into type A person's plasma? Definitely not. This antibody interacts with this antigen. Can AB give to B type? We already established. No. This is going to interact with this, and we're going to have agglutination. Can AB give to type O? Absolutely not. Both of these are going to interact with both of these, and you're going to have agglutination. And that person's going to clot, they're going to clump, they're going to clot. They could die if they don't get adequate medical care, or even if they are getting medical care, sometimes by chance these people die anyway. So why did I have to tell you all of that? Because agglutination is going to be the key to our blood type testing. Now, before we get to blood type testing, I do have to do one more thing. I have to add the RH factor. So it gets its own mini table, because there's only really 2 scenarios for RH factor. You can be RH positive and you can be RH negative. And the RH factor determines whether or not you have the surface protein known as the D-type protein. So you may be thinking, why didn't we just call it ABD? Or AD or BD if it's a D-type protein? That would have been nice. Yeah, it would have. We discovered it in rhesus monkeys. I'm going to erase that before you guys write it. We did experiments on rhesus monkeys to figure this out. That's why it's an RH factor. Okay, and it's also slightly unique compared to the rest of these. So you can have a blood cell that has the D-type protein. Lots of opportunities for inappropriate jokes talking about the D, but I'll try not to go too far into them for the purposes of this recording. It's for in-person class only. You can have the D-type protein. If you have the D-type protein, you are positive. If you do not have the D-type protein, you are negative for the D-type protein. You can have a positive blood. You can have a negative blood, and so on and so forth. B positive, AB positive, B negative, AB negative. So it's just an additional protein. The difference is in the plasma. Those who are negative are not automatically born with the anti-D antibody. You only get the anti-D antibody after you have been exposed to the D-type protein. There's that joke I'm not making. And that joke actually works pretty far, because how do you get exposed to the D? I'm trying so hard, guys. How do you get exposed to the D-type protein? Typically, the way a person gets exposed to the D-type protein is through pregnancy. An Rh negative mother is pregnant with an Rh positive infant. And at some point, There is a placental barrier, so mom's blood and baby's blood should not be mixing necessarily, but at some point during the prenatal testing periods or some kind of trauma or just the trauma of childbirth, mother and baby's blood is exposed to each other. And so that's how the mother who is Rh negative gets exposed to the D-type protein and develops the anti-D antibody. So this is an antibody that only develops on exposure, whereas the rest of these you're born with. That's why it's a special category. And of course, somebody, that's just a null, that's an Na, let's do Na, be consistent. Somebody who has the D-type protein, of course, we want the opposite for their plasma, so there will be no anti-D antibody in a positive person. Why is this exposure and development of anti-D important? Why does it get its own header? So imagine a time in history where we figured out A, B, and O. And we got that figured out. We started doing blood transfusions. A lot far fewer people are dying. Hooray! But every once in a while somebody dies. And we can't really figure out how until later. Or then actually, it's a little bit more tragic. This is going to get kind of upsetting. What about people who are experiencing their second pregnancy? An Rh negative mom, pregnant with her second Rh positive child. Before we discovered this, there was a risk of something known as erythroblastosis vitalis, also known as hemolytic disease of the newborn. Again, this is beyond our learning objectives and study guide right now, so I'm not going to spell that out. You don't have to write it down. I'm just making a connection. So a certain number of babies were dying, and we couldn't figure out why, and it was because after the first exposure to the anti-D protein, mom developed the anti-D antibody. On the second pregnancy, the mom's blood, upon mixing with the baby's blood, introduced a whole bunch of antibodies to the baby's blood and caused an agglutination process in the baby. And of course, we know that that can be lethal. So ultimately, after doing research on this condition, erythroblastosis vitalis, also known as hemolytic disease of the newborn, you can tell how many times I've had to say that, we determined the Rh factor separately. So again, this could apply to any of these. You could have A positive or negative. You can have B positive or negative, AB positive or negative, and O positive or negative. And that does impact our donations, right? So now all of a sudden, if you're O positive, so I'm going to do this in a different color because we're going to do an extra layer here. And I don't want you to assume it's part of the rest of this. So let's say you're O positive. So you have your square, we're pretending it's square protein, surface antigen, the D-type surface antigen, but no other surface antigens. And let's assume that this is a B negative person who has had exposure to a D-type protein in the past and has the antibodies. We're going to always assume the anti-D antibody is there to be safe. So can this person donate to this person? The answer is no. Somebody with a positive Rh factor cannot donate to somebody with a negative Rh factor. However, somebody who is negative can donate to somebody who is positive. So this is fine. This is not fine. Check mark. Okay, I think, oh wait, no, I gotta do blood type testing. One more thing, and I'm actually gonna do this on a new page. Okay, so hopefully you're looking at the new page. How do we test people's blood types? We take a few drops of blood. PowerPoint has 3. I'm going to do 4 because I'm a scientist and I like to have a control group. What we have developed as scientists is artificial anti-sera. What I mean by this is I can have an artificial anti-A antibody, anti-A, artificial anti-B, and artificial anti-D. Cool. These are anti-sera that not me personally, that scientists have developed that I would use in my blood type test. So we take the blood sample from a person and we drop a drop of blood into our first little well here, and our drop of blood into our second well here, and our third drop of blood into our third well here, and our fourth drop of blood into our control. And then we start introducing the anti-sera. In our first well, we are going to put the anti-A antibody. Hooray. In our second well, we are going to put our anti-B anti-sera. Sorry, antibody. Our artificial one, our anti-sera. And into the third well, we will put our anti-D antibody. And in our control, we will not put anything because this is our control. And if it messes up, we'll know we did something wrong. Now, I've done this in the lab. This is something that we do in laboratory environments, usually with sheep's blood. At my last institution, they let me do some actual students. We used student blood. Super fun. Okay. Now, if you put the anti-A antibody onto type A blood, you already know what's going to happen, don't you? So the anti-A antibody meets up with the A-type surface antigen. What happens to it? It agglutinates. So if somebody is positive for the A-type antigen, the result is that after a few minutes, this drop of blood, let's assume it's spread out into this entire well, it's all of it, is going to start to look clumpy. It's going to do something called agglutination. If it did not have it, let's do a hypothetical over here. If it was not in position of if it was not in possession of A-type antigens, it would just look like a drop of blood after that time has elapsed. So this is positive for, I don't want to use positive and negative, do I? So this means that they do have the A-type, and this means they do not have the A-type blood. Right? And we're going to see that pattern over and over. If you took artificial anti-B sera and you put it onto blood that did have the B-type antigen, you would see agglutination. So this is a type B. If you put it onto a drop of blood that did not have the B-type surface antigen, you would get a normal looking drop of blood. And if you likewise put anti-D antibodies onto a blood sample that did have the D-type surface antigen, it would agglutinate. And if you put the D-type anti-D antibody onto blood that does not possess the D-type surface antigen, it would not agglutinate. So let's do an example of this. And again, you didn't put any antibodies onto the control. So if this looks like this at the end, awesome. Yes, blood everywhere. Okay, let's try that again. I hope you guys enjoyed my metal voice just there. I'm glad you can't see me on camera. It's probably for the best. Okay. So control should look like that. If control looks like that, you did something wrong. The most common thing that people do wrong is, it does help to stir this at some point. You might take a toothpick and kind of move this around to get reactions going. Sometimes you might like take the toothpick here and then take the same toothpick here and the same toothpick here and the same toothpick here just because it's the control and you do the same things. And then that would cross-contaminate it. And you would take all these anti-sera and spread them across and all of these would agglutinate. So that's why you do have a control, because that is a common error that people make. So okay, down here, These wells will be in the same order. I don't even need to label these wells because they're going to be the same. Let's make sure I don't cover my whole screen with blood. Okay. So this is where we're putting anti-A, anti-B, anti-D control. So let's say your final result looks like this. Too much of a perfectionist. Okay. Final results look like this. This is where you put anti-A, anti-B, anti-D control. What blood type is this? This agglutinated and this agglutinated. When you put the anti-A antibody on anti-sera onto it, it did agglutinate. When you put the anti-B anti-sera onto it, it did not agglutinate. When you put the anti-D anti-sera onto it, it did agglutinate. That means this person has the A-type antigen and they have the D-type antigen. This person is A positive. So that's going to be your goal. You're going to need to be able to read tests like this. The most common problem people have identifying them is simply getting it backwards. Which of these means you do have the surface antigen, and which of these means you do not have the surface antigen. So I assume you're going to want to repeat that part quite a few times. Don't assume that you understand it. There's a learning curve. This does take time. And just email me and ask for office hours if you don't understand it or you're having a bad time.